Hello, everyone. Thank you, Mariam, for the introduction, and thank you to the organizers for giving me the chance to speak today. Today, I will be discussing why some microorganisms are classed as pathogens, so as harmful, while others are considered commensals or beneficial. And I will be doing this both in the context of historical discoveries of published literature and some of the research that I work on. And in particular, I would like to stress how the classification of pathogens and commensals is really dependent on the historical context in which they're being studied. So historically, infectious diseases have caused a significant number of deaths, and up until the 1940s, they were actually the leading cause of death. I found this graph comparing the top 10 causes of death in 1900 to 2010. And as you can see, in 1900, the leading causes of death were things like pneumonia, influenza, tuberculosis, gastrointestinal infections, whereas in 2010, they became things like heart disease and cancer. And what jumps out immediately is not only that the number of deaths from the top 10 leading causes has really reduced, it's almost halved, but also that the types of diseases that we're dying from have changed. They're no longer infectious diseases, particularly in the Western part of the world, but they are particularly so-called lifestyle diseases. And I should specify that this data comes from the US. And in some ways, it's true to call things like cancer and heart disease lifestyle diseases, but in other ways, it can also be quite misleading because it fails to address how far we've come in the treatment and in the prevention of infectious diseases. People were still dying from things like cancer and heart disease in the 1900s, but because in 2010 they're not dying from other ailments, these have become prevalent diseases. It shows also how far we've come in things like vaccines, regular screenings, antibiotics, and antivirals. Now, however, the past bacteria have been key contributors to mortality, and given the increase in antibiotic resistance, they are once again a pressing concern. To name a few examples, statistics have shown that 1 out of 7 people in the course of humanity have died from TB, from Mycobacterial tuberculosis, which is really impressive if we consider that the first sort of archaeological history that we have of people being infected with tuberculosis is from only 9,000 years ago. Another example is obviously Yersinia pestis, the bacteria responsible for the plague, that in the course of its major death, which was Black Death, close to 100 million people. And another example can be the cholera bacteria responsible for cholera, which killed 40 million people in the past 2 centuries. One thing that all these 3 bacteria have in common is that they were discovered during the so-called golden age of microbiology, which was a period of about 50 years where microbiology took huge leap forward. Now, of course, this was largely due to advancements in optical technology, in microscopy, and in assembly techniques. Bacteria had been observed long before that. The first record we have is from 1976, when the Dutch Antonie van Leeuwenhoek observed single-cell microorganisms in a microscope that he himself had built. He never officially published his studies, but he had regular correspondence with the Royal Society of London. He had several diagrams, several sketches, so it was known for several centuries that single-cell organisms did exist. But it was only with advancements in microscopy and in aseptic techniques that we were finally able to, sorry, got a bit distracted there. It was only with advancements in microscopy and aseptic techniques that microbiology really took things forward. This was largely due to the fact that single-cell organisms needed to be isolated as single monocultures for us to study them. And it was during this time that the causal agents of several leading diseases were discovered, and that the scientific community moved away from the miasma theory of disease, which was this idea that infections could be caused by bad air or bad vapors, or instead the germ fluids, the theory that microorganisms are responsible for disease. And it was during this time that techniques like filtering and observing microbes were developed, sterilization techniques came about. We finally started sterilizing the tools used for surgery, and also that the first laboratory-based vaccines were developed. And one of the cornerstone concepts that came about during this theory was the classification of pathogenic bacteria. So what makes a microorganism pathogenic? And for a long time, the straightforward answer was that it is microorganism that causes disease to its host. So Robert Koch, considered one of the founding fathers of microbiology, was able to isolate Bacillus anthrax, which is the bacteria responsible for anthrax. And by observing the life cycle of this bacteria and the way that it infected animals, he developed, together with his collaborator Frederick Loeffler, 4 parameters or postulates around which the microorganisms could be defined. And these were that the microorganism must be found in diseased but not healthy individuals. It must be cultured from a diseased individual. Inoculation of a healthy individual with the cultured microorganism must trigger disease. And the microorganism must once again be re-isolated from the infected individual. Now, these postulates are still relevant nowadays to some extent, but they're also slightly outdated. For example, we've all heard about symptomatic individuals in the past few years, so this would contradict the first postulate, or we know that people can have developed immunity or can have other microorganisms in their body which prevent infection from other microorganisms, and consequently this would contradict the third postulate. I think it's quite hard for us to fully understand what those decades were like. But this idea that this huge amount of bacterial species that were responsible for some of the most lethal diseases were all discovered in such a short time span really influenced the way that we think today. We discovered bacteria like tuberculosis, anthrax, the bacteria responsible for diphtheria, typhoid, the plague, all in a very short time span of approximately 50 years. And the scientific community went from not even supporting germ theory of the disease, not even fully contemplating it, to establishing parameters, postulates about which pathogen is defined as such. And one of the consequences of having discovered such important pathogens in such a short time span was that for several decades, the discourse around bacteria was centered around their negative impact rather than the positive impact. And it was only really in 1907, as we touched on this, that the first modern concept of probiotics really came about. And it came about with the Russian scientist Ilya Mechnikov, who, as Manon had explained, was very much interested in this idea that certain bacteria could be linked to longevity. He was also already really interested at the time in the idea of a unique intestinal flora in different individuals. He became fascinated by the fact that in the 1842 cholera pandemic, only some people were getting sick with cholera, whereas others were equally exposed to the infection but not developing any illness. And he's actually known for having described the sample himself and not having developed any disease. Of course, we've already touched on the fact that ancient civilizations used fermentation as a form of preservation of products, and the health benefits of fermented dairy products were already known. But it was only in 1935 that a true turn in the commercialization of bacteria came about, and it was with the isolation of Lactobacillus casei and the development. An interesting thing is that this concept of having some bacterial species be responsible for potentially fighting off harmful pathogenic species was already introduced by Ilya Mechnikov as orthobiosis, which can translate to right living or correct living. But it was only several decades later that the definition term probiotic came about, and it was introduced by the German scientist Werner Konrad, who defined a probiotic quite generically as an active substance that can be essential for the healthy development of life. And of course, we could delve deep into the history of probiotics, but this is something that Manon has already done. And what I would like to place our focus on is the terminology that we use to talk about the microbiome, because it has very interestingly evolved in parallel to major scientific discoveries. So, of course, in general, in the history of science, a lot of the science that we've conducted has been somewhat shaped by an inevitable anthropocentric approach that has been focused on human diseases. And this is particularly apparent in the context of bacteriology and the microbiome. So we use, of course, the identification of a lot of pathogens responsible for disease was driven by contrast to address deadly diseases. And the focus on probiotics came about because of this beneficial potential that these bacteria could have in improving human life, human health, and human longevity. Hence, we see the use of terms like orthobiosis or correct living and probiotic, pro-life. Now, in truth, probiotics are bacteria that have the potential to kill other microbes, to compete them, to fight them off. So we can only really call them pro-life in the context of human health, but not necessarily in the context of the pro-life or the pro-life. The definition of microbes as commensals or pathogenic is a bit reductive. It trivializes human microbe interactions and also microbe interactions. It also assumes a universal definition of health, which we know is. In recent years, there has been a shift from a more anthropocentric approach to a more microbe-centric approach. And the scientific community has accepted that more flexible and more accurate concepts than just good or bad, pathogen or commensal, are needed. And it has increasingly adopted an ecological theory to explain the microbiome. So we see the rise of terms like networks, niches, systems, or keystones in describing the microbiome. Furthermore, even though the discourse is still heavily focused on human health, we also see an interest in just the ecological maintenance of certain microbes. There is this idea that we are driving extinction of the microbiome because of our heavily processed and more Western diet. But this interest is not only focused on human health, but it's also starting to be appreciated from a broader ecological perspective. And in the last few minutes, I would like to stress how this shift in approach and focus and terminology is necessary in regards by bringing forward an example, which is that of the bacterial species that I work with. But before I do that, I obviously wanted to point out that since definitions are so inevitable and absolutely necessary for effective communication, especially in science, a new term has come about, which is that of pathobiont, which is a symbiote that is able to promote pathology only when specific genetic or environmental conditions are favoring the host. And for those of you who are not familiar with the concept of symbiont, it's an organism that lives in association with another. And this definition is in some ways really a step forward because it stresses the mutable nature of microorganisms, but in other ways it's still slightly flawed because it keeps putting microbes only in the context of human health. And it also assumes that there's sort of a threshold in normal genetic or environmental condition, which we know not to be the case. Now, as I mentioned before, I would like to stress the step forward in terminology by bringing forward the example of the bacterium that I work with, which is a type of Escherichia coli. And for those of you who are not familiar with E. coli, they are a really abundant, really diverse group of bacterial species. They have traditionally, like almost all bacteria, been classified as either pathogens or commensals, and pathogens are furthermore classified as based on the pathogen, so based on the location in which they can cause disease and the type of disease that they can cause. So there's some E. coli that can infect the meninges and cause sepsis, others infect the gut, others can cause kidney failures, others the urinary tract. But even though E. coli get a bit of a bad reputation, actually the majority of E. coli bacteria are what we classify as commensals, and they colonize the human gut shortly after childbirth, as early as 40 hours after childbirth, and they are lifelong companions. The bacterial strain that I work with is classed as commensal and it's called Commensal 1917, and it has quite an interesting history. So it was isolated for the first time by a German physician called Alfred Commensal. He was a military doctor during the Balkan War, and he noticed that in his camp, there was an outbreak of Shigella. Shigella is another bacteria that's associated to disease and fever. And he saw that only one soldier did not develop sickness. He was already interested in intestinal flora at the time. So he took a sample from the soldier and was able to isolate a bacterium that showed antagonistic activity against other bacterial species. And he had the scientific and the business acumen to study this bacterium further and to develop it into a probiotic called Multifaceted, which has really successfully been used as a probiotic for the last 100 years. It's used to treat various intestinal disorders like ulcerative colitis, or colitis, or inflammatory bowel disease. And there is really a lot of published literature in support of this strain as a probiotic. However, in 2006, a group of researchers in France found that Commensal also produces a powerful toxin. It's called the genotoxin because it can damage DNA. And this toxin is called colibactin. And by damaging DNA, it sort of breaks down the entire cellular machinery, causes cell cycle arrest, odd proliferation. And as you know, odd proliferation in cells is associated to the development of cancer. So Commensal 1917 has been associated to the development of colorectal cancer, and it's also found in 50 to 60% of colorectal cancer patients, whereas it's only found in 20% of healthy controls. So there is this really dual nature, this mutable nature of a strain that on one hand is a commensal, it's used as a probiotic, but on the other it also produces this toxin that is able to potentially trigger colorectal cancer. And I remember when I first started reading the literature around the strain that I would then continue my PhD with, that I was slightly puzzled and also a bit frustrated as to why we classify it as a commensal when it has this huge pathogenic potential. But after a while, I think I just accepted that maybe a definition of it, a classification of it as a pathogen or commensal was not really necessary. And this message was really driven home when a paper came out in 2022. And it showed that basically when you remove the genes that are responsible for the production of this toxin and this bacteria, it loses the potential for cancer, it loses the ability to produce this toxin, but it also uses its probiotic potential. So basically it becomes completely irrelevant to the context of human health. However, by not producing this toxin, it's also not possible for it to compete other bacteria. So it's completely relevant. In conclusion, our understanding and consideration of bacteria have been immensely shaped by the pathogenic potential and by the golden age of microbiology. In recent decades, we have appreciated that bacteria can be pathogenic, commensal, both, either, and our language is adapted to address this. It's worth conducting research from non-human perspectives. And this idea that for a century, we sold the probiotic, we advertised the probiotic that can also have this powerful cross productions for rectal cancer potential. It's worth taking into account that we still don't know about commercializing products. Thank you for your attention.